Hey, Hammy here, back with the second video of Chapter 12 over 12.2, on which we will look at DNA replication, in other words, DNA making a copy of itself. Remember in the last video, one of the things when we were talking about the genetic material is that the genetic material needs to be copied from generation to generation of organism or of cell. So it has to be copied with very high fidelity. This video here today looks at that process. Okay, again, replication comes from the root word replica, okay, which means an exact copy. Okay, so we need to take the DNA molecule and make a copy of it. It's semi-conservative. So if you remember, DNA was double-stranded. It actually runs anti-parallel. But what we're going to do is we're going to use each of the original backbones, split it apart, and use the exposed base pairs as a template to build the new base pairs on those exposed nitrogenous bases. So it will be exactly the same when we're done, but if you're taking your DNA strand and you're going to separate it, okay, pull it apart, and then you're going to build your new strands here. Okay, so you're half new, half old. That's where the term semi-conservative. You're conserving half of the DNA strand in each generation that this gets copied. Again, just a little bit better picture than I can draw. Here's our double helix DNA up here. Okay, and we see our base pairs C and G, A and T, our base pairing rules here down the middle. Okay, at the replication fork where it's being split, you see that if we pair a G with a C over there and a G over here and a G over here, those are bad G's, you'll see that when we get done, okay, if we compare that right here, if we compare that, okay, same. So what you see in blue here are the old or the parental strands, or what we sometimes would call the template strands. And then down the middle, okay, and this is kind of wound into that double helix, but these two would be the new strands that were built here. That's the semi-conservative nature. The actual process itself of DNA replication, first the double helix has to unwind, and then the two strands have to separate. That is done by, we'll learn a couple different enzymes here in this video. The one is DNA helicase. Okay, the helicase is the one that's going to be unzipping the two strands. In other words, removing the hydrogen bonds between the nitrogenous bases in the middle of the DNA. There are SSBs, single-stranded binding proteins, that attach to the newly separated DNA. This prevents the helix from recoiling up, kind of like it's holding it open, like a funny thing you wear at the dentist or orthodontist sometimes. Holds your mouth open, makes you look funny. Kind of these SSBs hold that DNA apart so that the enzymes can get in there and replicate or copy the DNA. Again, we use complementary base pairing between the new nucleotide and the old nucleotide that's on the template strand. So wherever there's an A on the new strand, it puts a T, and vice versa. And wherever there's a G or guanine, it'll put a C, or vice versa. Okay. And before it does that, a little DNA primase puts short RNA primers onto the DNA strand to be replicated. And then along comes DNA polymerase. Okay, that is the actual DNA builder. Okay, the DNA polymerase recognizes that RNA primer, gives it a starting point, and then begins DNA synthesis. The two strands are replicated differently. There's a leading and a lagging strand, and I'll show you this here kind of on the next slide. DNA polymerase can only build DNA in the 5' to 3' direction, because you remember on the last video we talked about the 5 carbons in the deoxyribose. We use that, and the two strands actually run anti anti-parallel to each other, okay, like a highway. And so the one side, they can just zoop, they can just read right down through. On the other side, they have to jump in, read back, and then connect them. Jump in, read back. Okay, and this takes a little bit longer, so we call it the lagging strand. And once it gets all the way done and all connected, the two strands separate, you've got two new daughter DNA strands that are exactly like the original one. This is just a good kind of a diagram or schematic of what's happening. Okay, here's the parental DNA helix. This is where we started. Here's some of your important enzymes. DNA helicase is unwinding and unzipping here at what we would call the replication fork. Our SSBs, our single-stranded binding proteins, are kind of holding that DNA open, keeping it from closing up. And then you have our DNA primase that's coming down and putting a little bit of, in purple here, this RNA primer. Okay, so there's the enzyme, puts a little bit of RNA primer, and then DNA polymerase. DNA polymerase can recognize that starting sequence, and then it starts to build. Okay, but notice you have a leading strand and you have a lagging strand. Okay, because as this unzips, remember we said they're anti-parallel. Okay, the one here, this would be 3', down here would be 5'. So building the new one, the new strand in green here would be being built 5' to this end right here, 3'. And RNA polymerase can just read right in that direction. On this side here, though, the original strand is 3', and out here is 5'. So our DNA polymerase cannot read that direction. So what has to happen, it can't go, it can't build 3' to 5'. It can't build this direction, okay? So what has to happen is the new strand here is 5' to the green end out here is 3'. So it has to jump in, read backwards, jump in, read backwards. And then when it gets done, there's another enzyme, DNA ligase, that kind of seals all these ends together. There are also enzymes that come along and remove the RNA primer and put DNA nucleotides in there. And this takes longer, and these little pieces or fragments are called Okazaki fragments, named after the scientists that helped discover this process and how it can actually go backwards on the one strand. Okay, so this would be the DNA replication fork, and this is sort of the structure, what's happening, but this is happening really, really, really fast. There are some differences between prokaryote and eukaryotic replication. Prokaryotic replication, remember prokaryote, we're mainly referring to our bacteria that do not have a nucleus. Their DNA is usually not in strands. They usually have a singular circular loop of DNA. Okay, so here's our bacterial cell, no nucleus, but then here in the center region, they just have this big coiled up DNA loop right here. Okay, big coiled up DNA loop right here. And so they just have to replicate or copy that one circular DNA loop. And what it does, it has a origin of replication, or it starts at one point and then moves in both directions around that point until it gets all the way around. Some of the most common bacteria, the Escherichia coli, that we do a lot of experiments on, replication takes about 40 minutes, but the cell can divide every 20 minutes. So you see we're in a predicament. The cell can divide faster than the DNA can copy itself. So actually, as that's dividing, a new round of replication will start at that points of origin before the other one is even finished, because the bacteria can just grow so fast. It has to keep producing new strands of DNA to keep up with that. This is just the visual of that. So here's that loop just kind of unwound and made into a simple circle. Okay, and then we have our origin, our point of origin, or where replication starts. And you'll notice that the replication forks will go in either direction until it gets all the way around to the termination, and then pop. Okay, you went from one copy of the DNA to two copies. Okay, and what it's saying, what we said on the last slide, some bacteria can divide so quickly that as this starts, this replication or origin of replication right here will actually start dividing again. These two will start dividing before it even finishes this round. And so it's just this continual process to provide enough DNA for that bacterial cell. In eukaryotic replication, most of the time we're talking about linear strands of DNA, often wound around proteins, histones, to make chromosomes. And so what happens, very similar in the loop where you had one, it went both directions. With a linear piece of DNA, you're going to have multiple points of origin along that chromosome, and it can unzip and it can break open right here, and it can read that way and that way. This one will read this way, and they'll all kind of form these little bubbles, and they'll all move outward until they all meet, and it'll pop, and you'll have two new strands of DNA. And again, we need our DNA polymerase as our main enzyme we're using for all this base pairing. And again, just kind of like I was saying, these replication bubbles can spread bidirectionally. So here's our chromosome. Here's our origins of replication. Usually there's a centromere at the center that kind of blocks what's happening. So you have someone, at least one on either side of that centromere. And then the DNA helicase will split the DNA, and you'll get replication forks traveling in both directions. Okay, so it started here, and it's going to move this way and this way. Started here, move this way, same thing here. And it's going to, these replication forks or bubbles are going to spread away from each other until they meet, okay? And they form the two new chromosomes. Something interesting there, that DNA polymerase is unable to replicate the ends. The ends of our chromosomes are called telomeres. They are copied by a separate enzyme called telomerase. Okay, DNA telomerase will kind of keep adding buffer to the end of those chromosomes, because every time you divide, you lose some of those ends. Unregulated telomerase is often we see in cancer cells and so forth, that will keep adding ends so that those cells never die, because the cancer cells divide, so they keep losing parts of the telomere. So they have an enzyme that they'll kind of mess up that enzyme, so it keeps adding those buffers so the cancer cells can survive. Last slide here is just kind of a difference between prokaryote, eukaryote. Again, so prokaryote over here, where we just have a circular ring. It starts at a point of origin, moves in both directions until you get two new daughter DNA molecules. Whereas in eukaryotic, you have more of a linear piece of DNA, and you have multiple points of origin and replication bubbles that move in either directions until you get our two linear pieces of DNA down here. Finally, we look at accuracy of replication. It's pretty accurate. DNA polymerase is very accurate, but it does mess up about once every 100,000 base pairs. But there are signals, and without getting too complicated, here you see, remember back to our cell signaling, cell cycle signals and stuff. Okay, if the DNA is damaged, we'll go through apoptosis, we'll stop the cell cycle. Things like CDK, cyclin can actually trigger apoptosis or DNA repair. DNA polymerase will actually go back and repair mistakes that might happen. So it ends up being, way less even than this. Very, very rare. And sometimes that mistake might be in an intron, a part of DNA that doesn't code for any protein or anything. So there might be a mistake and we would never even notice. But on a future chapter, we look at some of the mutations and stuff. We'll talk more about this and what happens when the DNA gets messed up or mutated. So that's it for DNA copying itself. In the next couple of videos now, we want to look at gene expression. How does that information on the DNA actually get made into or expressed as a protein? And so we'll look at the steps that are needed for that. This is Hammy, out.